Hello, everyone, and welcome to today's live broadcast, Fast Phenotypic Antibiotic Susceptibility Testing of Positive Blood Cultures, presented by Margie Morgan. She is a professor of pathology and laboratory medicine and also the scientific director of microbiology at Cedars Sinai Medical Center. I am Margie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Accelerate Diagnostics. For more information about our sponsor, visit acceleratediagnostics.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Margie Morgan. I will now turn the presentation over to her.
And thank you for having me here today. I'm going to take a little bit different tact, and I'm going to walk because some people might be bored by the laboratory, so I'm going to walk around and keep you all awake, okay? All right, so I'm going to talk about the actual laboratory participation in all of this, and we're going to be doing the actual testing in the lab. Blood cultures, yes, it's kind of complicated. We've been working on blood cultures for, 40 years, trying to make them more perfect, and we've gone from limiting contamination, which can be extremely important when you move toward molecular testing and rapid phenotypic testing, because if you have mixed blood cultures, it makes the whole situation a lot more difficult. So we're going to limit contamination. We're working on getting the adequate amount of volume in our blood cultures, because that's very, very important. And then let's move on. We need to get them to the laboratory and into the instrument as fast as we can. We need to pull the cultures when they become positive. In our laboratory currently, we're doing gram stains on positives. We're then performing molecular testing, as you can see, and communicating with our pharmacists about the results from our rapid molecular ID and resistance marker testing. But then that's when we slow down, and Dr. Grein and all of our crew, they become disappointed with us, because our past methods have always been to do very carefully make sure the identification was appropriate and do susceptibility testing using automated susceptibility systems. And even though maybe we only had one pathogen, it could take up to 40 hours. So that's where we were kind of letting down our stewardship team. So was this optimal for our patient care? Well, our pathogen identification really was quite rapid. We had identification by molecular identification methods. We also had MALDI-TOF that provided rapid methods. But sometimes we got into situations such as shown here, where we had a polymicrobial blood culture, where the resistance marker was detected, the Enterobacter cloaca was detected by a resistance marker, resistance and ID method, but it did not detect the Klebsiella pneumonia. That came out about 18 hours later. So it was like, surprise. Yes, we thought perhaps it only had an Enterobacter cloaca, but now we also have a Klebsiella pneumonia. So that is one of the limitations that Dr. Grein was talking about with some of these testing. It's very good for microbial calls, but sometimes you do get into situations using the best of these resistance and ID marker methods in mixed cultures. The other thing, is this optimal for patient care? Let's talk about the resistance markers themselves. Okay, these methods are very good, but they have limitations. The molecular marker, resistance marker detection from a positive blood culture comes out in 1 to 2 hours. That's quite remarkable. We're doing things so much more rapidly than we used to. But it can vary in its ability to properly guide therapy choices, particularly we found in our institution with the gram-negative rods. Detecting a resistance gene does not necessarily mean that it is being expressed. And as Dr. Grind discussed, some of the resistance that we are seeing in our institution, and I'm sure you as well, are not detected by these resistance markers that are on these panels, such as the AMP-C and the problems with porin and efflux. So we're doing all of these wonderful molecular resistance testing, but really, I hate to say it, sometimes the physicians are not very comfortable with some of the results, as he said, through our stewardship interventions, and they are really waiting for us for 2 or 3 more days to perform susceptibilities. So even though we instituted all of this new thing and we thought we were doing the newest cutting edge work, perhaps we really weren't supplying what everybody really needed. This is just an example of one of the discrepancies that we had just recently with our resistance marker testing versus our full phenotypic susceptibility. You can see on the left-hand side that our resistance marker detected ESBL, no ESBL on Klebsiella pneumonia. But if you look at the susceptibility result that we had up to 36 hours later, it truly was an ESBL. Now, what problems do you get into the laboratory with doing things like this over time with increasing percentages? Well, of course, there's the patient care issues, and we're greatly concerned with that in the laboratory. But also, we're greatly concerned that some physicians just never forget if you have one of these. And so it makes it a lot more difficult on our stewardship group to go back with more interventions to try to de-escalate antimicrobials, because perhaps the physician they're talking to was the one that actually had this patient that this occurred with. And so I think that's one of the shortcomings that we're seeing in the laboratory with just doing the molecular resistance testing. So is there a need for doing rapid susceptibilities? Well, there is for phenotypic antimicrobial susceptibility results are often necessary for very good optimization of patient therapy. The molecular platform can detect the presence of known resistance genes, but as we've shown in our laboratory, no molecular method can determine whether an isolate is susceptible to something, and oftentimes that's the question that they get with their interventions. Thank you. You're trying to explain to me what that CTX-M means. Now, what's it susceptible to? Are you sure this is what it's going to be susceptible to? And so that is perhaps not being provided when we're just doing the resistance markers, of course. And of course, we're seeing lots of new organisms coming through our medical center. It's amazing some of the organisms that we've seen over the last 6 months or so, and phenotypic results are certainly more helpful with this new emerging resistance in the gram-negative bacilli. So let's talk about rapid susceptibility results and possibly what the Accelerate Pheno system can offer. Now, there's been methods to speed up ASD testing over time, some very creative methods being done in laboratories. But the first FDA-cleared rapid susceptibility method was when the FDA granted clearance to the Accelerate Diagnostic Pheno Test blood culture kit. And that's what's shown here. This is the cartridge that's used for testing. It's truly a unique and divergent susceptibility method, and I will explain to you in the next few slides. And it provides rapid, final susceptibility testing in 7 hours compared to what we've been doing for 36 to 40 hours over time. So on the left-hand side was what our current method was. You can see it's many multiple steps. We thought we were doing cutting edge, the best of microbiology we could, and for the time, yes, we were. But by going through the left and all those multiple steps, it took us up to 40 hours to get a susceptibility. With using the Accelerate system, we go to this on the right-hand side. You can see there's less steps in the process, and so it's much more improved efficiency type of testing system, and we can provide a susceptibility within 7 total hours after the inoculation into the system. So we really think we're not only doing a more rapid job for our physicians and for our stewardship pharmacists and individuals, but we're also more efficient in the laboratory in the way that we are testing. So this is the Accelerate Pheno system. Some of you may be quite familiar with it, perhaps some of you not. Over on the left-hand side, on the top, is the system. It can go 1 to 4 modules. I believe it can even go up to 8 modules per PC. And you can see the testing modules, and there's also a touchscreen monitor that comes with the system. The module, which is in the middle, if you opened up that module and took a look inside, you can see it's a very complicated and very sophisticated instrument. This provides an automated pipetting robot with a digital camera and a custom microscope. So we're getting into what's so unique and divergent about this system. As you can probably tell, I've been in microbiology a lot of years, and I've been doing susceptibility testing since the early 1970s. And forever we've relied on, is it turbid? Is there in spectrophotometry readings? So this is what's truly divergent about this technology. We're moving away from just turbidity, and we're actually making use of newer digital camera technology and the custom microscopes to be able to look at the organisms themselves. And that's what's unique. The kit, you can see it's a plastic, over on the left-hand side, that blue circular thing is the flow channel cassette. The white is the reagent cartridge, and that little vial there is where you put the blood specimen. So you have your positive blood culture that comes out of your automated blood culture system. It is then aliquoted into that small little plastic vial and inserted into the cartridge and put into the system. Within the system, it's going to lyse the RBCs. It's going to filter out all the RBC remnants and other particulate matter in the specimen, and it's going to immobilize the bacteria onto a grid. After immobilized, the first thing that happens is the identification of the organism that is present in that positive blood culture. Okay, and it does that by fish probes. That takes approximately 1 hour and 20 minutes. The next step, of course, is the performance of the MIC. I'm sorry if I'm standing in front of you. I will come over here for a while. The next thing is the performance of the MIC, and that's what I think is really so fascinating and different. And we're going to talk about that on the next slide. The susceptibility is done not by turbometric methods that I've been doing since the 1970s. It's done by morphokinetic cellular analysis. In studying these organisms, it's a totally different way of looking if an organism is resistant to an antimicrobial. It starts with tracking growth rates and establishing a growth curve. And then it will look at the division rate distribution of the organism. So instead of getting turbidity, you get very, very sophisticated looks and snapshots of what the organism is actually doing as it grows in the presence of those antimicrobials. So this is just a very, very different, unique approach to susceptibility testing. You can see this one is a resistant organism. It just continues to grow and beautifully repopulate. And then on the right-hand side, you can see that the E. coli is susceptible and it is dying off over time from being exposed to the ceftazidime. So that's what makes this system so unique. This is what the cartridge, where the cartridge is put into the testing module. And this is where the cartridge contains the blood sample and is put into the instrument. There's one cartridge for all testing. Whether you have a gram-positive or gram-negative isolate, it goes into the same cartridge and put into the instrument. This would be the organisms that would be identified, if it were a gram-positive organism, identified by the fish probe. And these are the yeast, the albicans and glabrata. And these are the antimicrobials that are tested on the test cartridge if you have a gram-positive organism, which is identified. If you have a gram-negative, it will identify one of these 8 organisms by the fish probe, and these are the antimicrobials, which will be tested for the gram-negatives. Okay, so does this work? Well, yes. One would assume that it works. It cleared FDA, so of course it is past the ultimate test, and it has very good correlation with broth dilution methods, which it was tested against. So, and this is clinical trial data at the top and listed over here on the right-hand side. There's 1,800 positive blood cultures that was originally studied. The results for identification were available in 1 hour and 20 minutes. You can see all of the organisms that are identified on this panel and each of the sensitivities and specificities. Overall, in the clinical trial, the sensitivity was around 97.2% and the specificity 99.4%. We have a much less experience. We have not done 1,800 samples yet. In our experience, we've done 85 positive blood cultures so far. Results were available for ID in 1 hour and 20 minutes as found in the clinical trial. Our overall sensitivity has been about 97.6%. We've had 2 Klebsiella that were not detected. And our overall specificity was 98.8 with a confusion of 1 culture between Enterobacter and Klebsiella. These are the susceptibility results from the clinical trial. You can see once again a very, very high essential agreement and categorical agreement for the susceptibility results. Once again, 1,800 positive samples with an overall EA of 95.6% and 95.4% for specificity. The FDA demands greater than 90% for acceptance of a system into the market. Yes, this might not be the end-all of all antimicrobials that you might want tested on some of your organisms. You might have, on occasion, have to do some offline testing, and I'm sure that Accelerate will be doing their best over time to add some of these newer antimicrobials to the test panel. But for right now, these are the ones that are currently available. This is our susceptibility results at Cedars Sinai. We match very favorably with the clinical trial, and we have not had any issues with formulary antimicrobials failing on the Accelerate system. Okay, how about monomicrobial versus polymicrobial calls? I was talking earlier that there was an issue sometimes with our molecular systems in not being able to tell us if there was a polymicrobial issue. Okay, so what about monomicrobial calls? We're very fortunate that we have a very low contamination rate in our blood cultures, and so most of our blood cultures, not all, but most are monomicrobial. And so that makes this system extremely useful for us. Okay, so for a monomicrobial sample, it is positive for only 1 pathogen, of course. The positive predictive value is 99.6%, and if you have with a gram stain interpretation, if it's 97.8 if you're bypassing gram stain and just doing direct inoculation. We've had very few failures, and there were very few failures in the clinical trial of monomicrobial calls. The only thing that you could get into problems with if the numbers of organisms in the blood culture itself is so low that it's not being able to be detected by the system. How about polymicrobial capabilities? That was one of the issues that we saw with our molecular resistance markers, is they were not able to tell us most of the time if there were more than 1 organism. That's one of the unique features of the Pheno system, which I think is very useful. Even if it's not able to identify a second organism or so in the blood culture, it will give you an alert that there is a second organism present. So you will never get into the situation like I had in that first slide. Okay. So you can ID up to 3 ID targets on the panel, but if it's a polymicrobial with on and off panel, it will report the ID target organism, and it will alert you that there is another organism present on the panel. And that can help very much with our stewardship efforts. It will do the MIC on the predominant organism. If there's not an organism in predominance, it will not provide you with a susceptibility, but you may provide you with organism identification. And then you would have to do offline susceptibility testing. If the 2 organisms that are present are so close in the way they're growing and in their division, it will not supply you with a susceptibility. As I say, we've been lucky in our experience at Cedars is we have mostly monomicrobial blood cultures. And so that has not been a big issue. But every medical center will differ in that. This is the Accelerate Pheno report that you can see on the right-hand side here. It's easy to read. It can be on the screen, or you can print the report. It's an intuitive computer system, very easy to use, and it has customizable MIC interpretation for FDA, CLSI, or UCAS breakpoints. It has export rules for interpretation. It provides assay analysis, as you can see here. There's notes that provide you information for interpretation of the report, just like the current AST systems do. It's certainly interfaceable, and the people at Accelerate will help you and provide assistance in interfacing this instrument. What's getting started? We met with stewardship, of course. They decided that the best and most clinical value use of this test would be for the gram-negative rods. I think I've made it clear to you that we've had some failures with such things. Accelerate will offer a verification protocol. It's about a 25-page document that walks you through the verification process. I can't tell you how much that helps a microbiology laboratory get started. They also have developed an individualized quality control template for microbiology laboratories so we can establish weekly QC. I think one of the most fascinating and wonderful things about this system is the QC. It's a frozen little snap-in module. No more 0.5 McFarlands. No more organisms all over everywhere that you have to accumulate every week in order to do QC. It's a very, very unique and very time-saving way to do QC. And if you are going to do an LIS connectivity, Accelerate will assist you, and you certainly need to allow time for that. It always seems like the microbiology laboratory moves a little bit faster than does those computer people. Okay, finally, why use the Accelerate system? Well, of course, it supplies rapid phenotypic susceptibility to improve patient care. Of course, it is accurate. The FDA and all of the clinical trials have proven that. We have found that there's limited clinical utility of gram-negative resistance markers, and that we get good results from our gram-positives, but Accelerate really does improve our therapy and stewardship interactions with our gram-negatives. You can replace your resistance marker testing by actually doing rapid final susceptibility testing. And in our high-risk patients where we're starting to see more unusual organisms, we really think this will be very, very useful. And our stewardship team was in great acceptance, and they're very happy that we're moving in this direction. And they said that their interventions will be more definitive with a final susceptibility that includes susceptible antibiotics and not just resistant markers. And it's worth that 7-hour wait to get that information. And with that, Jonathan and I will entertain any questions that you might have about the system. Thank you.
Thank you, Dr. Morgan, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what advice do you have for a lab director to gain alignment from her or his organizational leadership to move forward with implementation of new diagnostic equipment?
Well, that's a very interesting question. I guess the first thing I would say that you need to do your homework. Is the diagnostic equipment really going to offer something of value to your hospital, to your stewardship group, to your group of physicians and patients that will be using the information? I would go to the literature and see if there's any literature on the topic. If I'm attending a meeting, I would make sure to drop by the booth that is actually selling that equipment and see if I can see it firsthand. And it always helps to talk to users if the equipment is out in the field yet. So will the results from this instrument be useful in your institution, and particularly with your own patient population? That would be very important. Is it possibly worth the increased cost that the laboratory may have to bear? Sometimes there will be offset savings to the hospital, but after all, we're going to have to bear the brunt of the load in the laboratory, so I'm going to have to provide justification for why we should do so. Collaboration could be very important. Stewardship committee members, pharmacists, infection control, and ID physicians. It's going to be a collaborative effort, and they can help me provide necessary data to administrative leaders. And I think the most important thing is for years we've been talking about things, but I think we're to a point now where we really have to provide data. It's not just words on how this equipment can really help us with either antibiotic optimization, patient days in the hospital, or help save lives or lower C. diff rates. We really need to provide data to prove that this could be true. Also, we need to provide potential budget issues for the laboratory, but also how this could save money for the hospital. Perhaps show literature, if available, how this has worked out in other institutions. Also, do you have staff ready and able that you can do this technology? Are they technically able to do the verification studies and all the patient testing? So if we have to make an argument to administration for increasing staff, that could be very, very difficult. So I need to show data that the equipment can be used for existing staff, of course, if that is true. So hopefully with all this information and collaborating with others, we can make a valid business plan and get an instrument like this implemented even in my hospital, Cedars Sinai.
Thank you for that answer. Our next question is, if you were in an institution that did not currently have resistance marker testing, would you utilize the Accelerate Pheno TM system for gram-positive blood cultures? And what other circumstances would you recommend using this device for all positive blood cultures?
That's a very interesting question. If you lack resistance marker testing in your own institution, I think one should certainly look at the use of the Accelerate for both gram-positive and negative infections. We did not do it in that way because we were actively using another system for gram-positive markers. In our institution, the gram-positive markers had been used for years and were used successfully. However, in some institutions, that might not be the case with resistance marker testing. And studies have indicated that clinicians are often unwilling to de-escalate to optimal antimicrobial therapy until antibiotic susceptibility is actually obtained. So I think there's certainly an understanding that it would be good to move to Accelerate for both gram-positive and gram-negative applications. There was a paper recently published in IDSA publication for a community hospital. I think it was the University Health of Augusta, Georgia. And in this situation, they showed case studies where having the Pheno test blood culture results really did help treat patients more than 50 hours faster than their previous methods. And it was demonstrated that having this fast AST versus just ID alone for the gram-positive bacteremia had a very positive outcome in clinical outcomes of the patient and really helped in optimizing therapy. So I think one could make the argument in the lack of any type of resistance testing or available information that it could be very, very useful to use the Accelerate for both gram-positive and gram-negative infections. That's being said, you do need to look at the gram-positive panel and realize there are potential, some small but potential issues, such as speciation of the streptococci with the gram-positive panel that had moved some of us into just using it for the gram-negative application. But I still think that it has a very strong use for the organisms that do exist on the panel. So I could see that institutions moving into using it for both gram-positive and gram-negative.
Thank you. Our next question is, what have you observed about clinicians' willingness to adjust therapy based on phenotypic MICs as compared to resistance marker test results?
Well, as I said in my talk, we're mostly using the Accelerate system currently at Cedars for the positive patient blood cultures that have gram-negative organisms. So in our institution, we went that direction because the clinicians felt more comfortable using, they did feel comfortable using the gram-positive resistance marker information, but did not feel comfortable using it for gram-negative bacilli. Our gram-positive resistant markers have been pretty reliable for the MRSA, the VAN-A, and VAN-B, but they have not been as helpful for the gram-negative infections. And we've had failures with our gram-negative markers on the current system that we're using, such as the CTX-M, the VIM, the IMP, and so forth. It wasn't necessarily a failure of these tests. They just didn't have the type of resistance marker or a capability of detecting the resistance that we're currently seeing in our gram-negative rods. We're starting to see some emerging resistance of AMP-C, porin, and efflux alterations. So it is really becoming very clear in our institution that there was a lack of confidence in just using resistance markers, and that they really wanted to have a susceptibility to be moving forward with changing antimicrobial therapy. And that's where the Accelerate has become so important and so helpful, is we can provide that rapid final susceptibility on our gram-negative rods and can move away from the use of the resistance markers for the gram-negative bacilli.
Thank you. Our next question is, please explain how the monomicrobial call provides clinicians with the confidence to de-escalate antimicrobial therapy. What is the significance of this?
Well, the monomicrobial call is cited under the notes on the report that you get from after the completion of the Accelerate run. It is actually on the report that you observe on the computer or when you print out the results. The monomicrobial call gives you information that that positive blood culture that you just ran on the system is positive for only one pathogen. The accuracy of this claim is in numerous studies, and it was cited in my report that I just gave, my talk that I just gave, that it's 99.6% positive predictive value with a gram stain interpretation and 97.8% positive predictive value without a gram stain. So the crux of the matter is that this call can help the stewardship pharmacists with their interventions to assist with antimicrobial optimization, and can help the physician to be more confident that there's not another organism that's lurking in that culture, that they can trust the results that we've produced. So the monomicrobial call is a very, very useful thing to have available to us in the laboratory.
Thank you, Dr. Morgan, for that answer. Do you have any final comments?
Well, I would just say from our experience in using the Accelerate system, it has really been very useful to our stewardship committee in moving forward with optimization of therapy. And as I've said in some of the answers to these questions, as well in my talk, it's been very useful, particularly for the gram-negative bacilli that we're seeing currently in our patient population. Unfortunately, we're starting to see emerging resistance of the AMP-C and porin and efflux alterations, and having a rapid phenotypic susceptibility has really proven to be very, very helpful.
I would like to once again thank Dr. Margie Morgan for her presentation. I would also like to thank Accelerate Diagnostics for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.